The first term which we will be discussing will be operon. Operon is the operating system. Operon. It is the unit made up of linked genes which is brought to regulate other gene responsible for protein synthesis. Operon is a set of genes which are, you can say, the master, which are regulating other genes and telling them, okay, now you can synthesize protein, okay, now you need not to be synthesizing protein and stop functioning. So that whole concept is operon. This whole concept of switching on and switching off comes under operon model. Clear? Other term will be cistron. Cistron is the segment of DNA which is actually being expressed. The segment, the structural gene, the cistron is the structural gene actually which is coding for certain important polypeptide which has to be formed. Cistron is the major structural protein. It has to be expressed. And who regulates cistron? It is operon. Operon regulates that, okay, now cistron has to work, his cistron has to be expressed, and okay, now we have enough amount of substrate present, so cistron has not to work. It should stop functioning. It should be switched off, and now we need this product, so it should be switched on. So two important things: operon is the operating system, or operon is actually the regulator, monitor, that okay, this time the cistron should function, and operon also governs when the cistron should not be expressed. Clear? So these two are important points here you need to understand. Further in this chapter we shall be discussing about all these two types of operon concepts. The first concept will be inducible operon. Inducible operon is the same, I told you just now, induction takes place in inducible operon. And the second one will be repressible operon, in which the repression, the stopping, no synthesis, turning off of the cistron takes place. Clear? So these conceptual thing I have told you today, and in the next class I'll be telling you about what actually happens and how the process is being regulated, what happens in the bacteria which makes or governs the synthesis of certain proteins in the particular Escherichia coli bacteria. So here in this whole chapter we will be studying only about Escherichia coli, because that eukaryotic thing is not in your course. See, if you have seen the notes, here is wobble hypothesis been given here at the end of this chapter in the notes. Yes, have you seen? So here in wobble hypothesis, I have already explained wobble hypothesis in the Zoom meeting, but because of the technical something problem, some of you cannot hear it properly. So let us discuss wobble hypothesis today again. So wobble hypothesis, it was given by Crick, and it says that a single transfer RNA can recognize more than one codon. Single transfer RNA can recognize more than one codon because more than one codon code for same amino acid. Okay, and then so that even if the slight change due to mutation in the codons, there is no change in the amino acid. How does it brought about? How it is brought about actually? The first, they are generally reading the first two codons. If the first two nucleotides of the codons they are reading, so if the first two nucleotides of the codons are same, the triplet will be coding for the same amino acid. Okay, if we see the example, UGU, UGC, UGG all code for arginine. Although you can see only first two bases in UGU, UGC, UGG are same, but they all code for the same amino acid, that is arginine. So this was wobble hypothesis, which says that the transfer RNA is one transfer RNA can recognize more than one codons, because actually more than one codon is having, see, there are 3 codons I told you in example. So these 3 codons can be recognized by a single transfer RNA, because ultimately there is a change only in the third nucleotide. So what will happen? Seeing the change only in the third nucleotide base, all these will be recognized by a single transfer RNA, and that single transfer RNA will be carrying arginine, because the message these codons have hidden in themselves is for the arginine. Clear? So for today, I think this much is okay, this much is clear to everyone. And just read the conceptual thing. Do not read the induction, inducible operon or repressible operon. Just read on the introductory part. If you can find it in your bio book, just read on the introductory part from there also, and understand the wobble hypothesis. In the next class, I shall be telling you about the mechanism of lac operon, mechanism of lactose operon, or what we call as inducible system, and the mechanism of repressible system. Clear? So let us wind up for now. Thank you so much.